Hello. On behalf of Nuvisan, I would like to welcome you to our talk today. I'm going to talk about how to safely reach proof of concept in early clinical development. More specifically, how is a first-in-man study planned so that all safety aspects are taken into account, and what are good strategies for the subsequent early clinical development program with a focus on safety, and how can a clinical proof of concept be achieved at an early stage. But before we explore today's topic in more detail, I would like to briefly introduce myself. My name is Wolfgang Timmer. I'm a medical doctor and a clinical pharmacologist, and I'm working as medical advisor at Nuvisan. I act as principal investigator for clinical trials, but I also give advice to our clients regarding the preparation of study protocols and clinical development plans. My personal work experience started 29 years ago. I had several positions in the pharmaceutical industry and CRO business with a focus on early clinical development. I cannot proceed to the next slide. Now it's working. We apologize. Before we start, some important notes up front, so-called house rules. Your microphone is muted by default. If you have a question, please use our Q&A box. That means not the chat box, please use the Q&A box. Type in your inquiry, and after the talk, I will endeavor to answer as many questions as possible. Please also note, this session will be recorded and will be available on our website, and you can find it in the events category. Who are we? Nuvisan is a fully integrated contract research and manufacturing organization. We are more than 1,000 employees distributed over 6 locations, and we offer services over the entire drug development chain, starting from target identification until the conduct of early clinical trials in healthy subjects or patients. The part of the development program I'm talking about is located in the transition from preclinical to clinical, as well as in the field of early clinical trials. The conduct of clinical trials is accompanied by clinical laboratory services and bioanalytical services, which are also offered by Nuvisan. It is important to detect safety signals at an early stage and to carry out exposure control, which also serves to determine the clinical dose for later patient studies. I would like to invite you for a poll. You should see a window opening and you can select different answers. This is a simple question. Have you worked with Nuvisan before? If you are not involved in outsourcing activities, you do not have to answer here. Now I can see the results. The majority of you have not yet been working with Nuvisan before, but this may be subject to change very soon. So I proceed with my talk. Yeah, I'm sorry, now I can't continue again. Now it's blocked again. Now it's working again. We apologize. Here you can see today's agenda. You have already heard about our services. Then I'm talking about, in the first step, about those considerations for first-in-man clinical trials and risk aspects, more specifically about design elements of early safety studies single ascending dose trials, and multiple ascending dose trials, which are first-in-man studies in a broader context. Then we have a look at the search for pharmacodynamic endpoints, abbreviation PD, and possible surrogate markers, and early proof of concept studies. And finally, we have a question and answer session. First-in-man trials are phase 1 clinical trials in the narrower sense. I would like to differentiate a little here, because when we talk about phase 1, phase 2, phase 3, usually the term phase 1 means phase 1 in a broader sense. That means all clinical trials in healthy volunteers, which also includes a variety of pharmacokinetic trials or trials to investigate the formulation or drug-drug interaction trials. Here we talk about the first clinical trial that is conducted in healthy volunteers in order to assess safety and generate information about exposure data. What I'm talking about here is a development concept for a classical investigational compound that binds to a receptor and that follows a dose-response relationship with regard to pharmacodynamic effects and possible side effects. Clinical development programs in oncology are fundamentally different. For chemotherapy drugs, first-in-man trials are usually carried out on patients and not on healthy subjects. There may also be different approaches for investigational compounds that are developed against rare diseases or when there is a high medical need. So when we talk about the classical approach, the design elements of first-in-man clinical trials are the following. There are different ascending dose groups, so-called escalating doses, with different subgroups of healthy subjects participating in this trial. If it is the first-in-man study and a single ascending dose study, every subject will only be receiving one dose. There are usually 6 to 9 dose steps. And then this trial is followed by a multiple ascending dose study, which has less dose steps compared to preceding single ascending dose study or single ascending dose part, because quite frequently the SAD part and the MAD part is combined in one study protocol. In the single ascending dose part, there's a placebo control, but it's not a one-to-one randomization. A common ratio is 3 subjects are on active compound and 1 subject is on placebo. The primary objective of the single ascending dose study is the identification of the highest dose that is safe and well tolerated after single dosing. The first single dose study is followed by the multiple ascending dose study, which confirms the safety and tolerability of all doses under steady state conditions. Usually, higher plasma exposure is reached when an investigational compound is given repeatedly. This depends on its half-life and of the dose interval. In this context, the investigation of the pharmacokinetics is important, not only for safety reasons to control the systemic exposure, but also to investigate the pharmacokinetic properties of the drug, which may be important for the determination of the clinical treatment regimen. In addition, data on pharmacodynamics are collected if this is possible in healthy subjects. In this context, I would like to mention the following. If you are attending a seminar today that is entitled How to Streamline Your Clinical Development, then it is often recommended to combine several studies in one protocol, a so-called umbrella protocol or integrated protocol. I would be cautious here. Especially in early clinical development, exploratory studies generate hypotheses that are needed to plan the following studies. For example, you have to know the pharmacokinetics of an investigational compound in order to be able to plan the blood sampling times in the next study. Or another example, the dose steps to be tested in the first repeated dose study are often only determined by the results of the single ascending dose study with regard to tolerability, exposure, and bioavailability. On the other hand, it makes sense to combine certain questions in one clinical study. For example, the investigation of food interaction in the first-in-man study is usually always helpful. I just mentioned every subject will only receive one single dose. The food interaction is an exception in this case, a particular subject will receive the compound once in fasted state and once in fed state. Now let's talk about the estimation of the so-called maximum recommended safe starting dose for first-in-man clinical trials. I am referring to the FDA guidance document here, which is state-of-the-art. You will find this FDA guidance in the internet. The basis for the determination of the doses in a first-in-human clinical trials are non-clinical data. Non-clinical data in terms of pharmacological data, you may have an idea what the systemic exposure should be to achieve pharmacodynamic effects, but also the pivotal toxicity study in relevant animal species to determine the safe dose range when the first clinical trial in humans is planned. Usually there is a toxicity study in one rodent species and in a non-rodent species. And in these preclinical studies, the so-called no observed adverse effect levels are determined, the so-called NOAELs, which is a milligram per kilogram dose. And this dose level is defined as the highest dose that has not produced any toxic effects. So it's the next lower dose under the dose that produced toxic side effects. Of these two studies, each NOAEL is converted to a so-called human equivalent dose based on certain scaling factors, which you can find in this guidance document. Every animal species has a certain scaling factor. This takes into account body surface area, but also some empirical factors regarding the metabolism of these animals. And then the human equivalent dose, the lowest dose is taken, and then another safety factor is applied, which should be at least 10. And then you get the maximum recommended safe starting dose, which is still a milligram per kilogram dose. Very often when there is a first-in-man clinical study, all subjects receive the same nominal dose. So when the study protocol says the body weight should not be below 70 kilogram, then this milligram per kilogram dose needs to be multiplied by 70 kilogram body weight. And then this is a safe starting dose for the first-in-man clinical trials. That means this is the lowest dose. And starting from this dose, the dose can be further escalated in your single ascending dose study. For certain drugs, it should be considered to replace this dose by a so-called pharmacologically active dose, which may possibly be lower. This term, pharmacologically active dose, is the American term. The European expression is MABEL. I will talk to that in a minute. When should an increased safety factor of more than 10 be applied? This may be the case when, on basis of your preclinical data, there may be a steep dose response curve or severe toxicities or toxicities without advance warning, non-monitorable toxicity, unexplained mortality, or irreversible toxicity. So these 4 points, severe toxicity and irreversible toxicity, are generally critical because there may be a higher risk in your first-in-human clinical trials. It's clear when there is non-monitorable toxicity and you further escalate the dose, then sooner or later you may run into problems. So if there are such significant issues, it should be discussed whether this drug should really be developed. Of course, this may also depend on the medical need. Steep dose response curve is an issue that is often seen in preclinical development. If this is the case, or also variable bioavailability or nonlinear pharmacokinetics, this may be a reason to investigate more dose steps in the single ascending dose studies and have narrower intervals between the dose steps to be on the safe side. The last points here, novel targets or animal models with limited relevance points into the direction. This may be very innovative drugs where you have limited information. This may also be a reason for concern, maybe a reason to be more cautious in the first-in-man trial, which may lead to a situation that only one subject is dosed on the very first day. Nevertheless,